So let's get going.
Let's start. We have seen some of the work in the past episode with the chart. The title of the presentation today is Deep Green, Bio Design in the Age of Artificial Intelligence, which is also the title of the book that Mark and myself are currently writing or completing, and that will be published in February 2023. I represent the academic side of the partnership. I have a double professorship at the Bartlett UCL London and at Eastbrook University, where I deal with urban morphogenesis and synthetic landscape. We have worked with a lot of microorganisms, insects. We have a lab in Innsbruck, and while in UCL we develop more the computational aspect. But here we want to show what we have developed in the last couple of years, which is we try to understand how this microorganism or bacteria, fungi could be scaled up to remetabolize some of the byproduct of the urban sphere. What normally we call pollutant, that is dangerous for us, but can become food and resources for other organisms. Here you see a project that we developed, also in collaboration with one of the scholars here in Kamchatka. They look at the remetabolization of heavy metal in the soil and water through the integration of a material system that contain microalgae and mycelium, and let's say a thick form of architecture that becomes almost geological.
Yeah, I guess this is the possible proposal for the line, if you want. Now, we were discussing today about the line, so we thought that this project actually is interesting because it's talking about something very large, and it's talking about the soil or the ground becoming in some way thick and perhaps also inhabitable. And I think in a similar way as Mitch was pointing out, we probably started this project from let's say the perspective of other species, and then through the use of computational processes we have tried, or the students in this case, the researchers have tried to extrapolate what could be like at a scale that is inhabitable to humans. And I mean, I think this concept of the thickened ground or inhabitable ground or ground scraper, I think there was a book from Ken Young, I think, which was another pioneer of environmental design, Malaysian architect Ken Young. It's not new, but I think what we were trying to propose here is the possibility to see these as a metabolic engine, right, that can perform not just at the microscopic scale or the human scale, but at some point become really a kind of geological force almost, right?
And our bacteria and microalgae can become a binder for soil and elements that are present inside to create a novel form of architecture. This has been applied to other contexts, and we would like to show just another small example. This is in London. Look at London in 100 years, and in particular at the River Thames, and deploy again a similar material system to the previous one involving biocementation, and look at how, if we accept that the east of London will be flooded according to the prediction, that how can we terraform this area to regenerate it and create a new architecture. We can see the video.
So yeah, this project again, it's part of the research in the urban morphogenesis lab, and it's very much related to water and the wet landscape, which we thought it was relevant to bring up today. I think there is also an audio, so we will.
In particular, also for the scholar that are looking at the liquid. See.
With the rising temperatures affecting the ozone layer, the glacial flows of the world are melting, which coupled with human growth and evolution is affecting biodiversity. This human and non-human invasive coexistence is having side effects such as COVID-19. Since the humans started to invent biodiversity's habitat, the boundary between entities was lost. In order to reduce zoonosis and biodiversity loss, an established boundary with nature regeneration is needed. With the melting glaciers, the sea level rise. Therefore, we are projecting a flooding London that surpasses the current barriers in 200 years. With this flooding, the new water volume would generate marshes at the edges. A marshland eventually works as a biodiversity generator. Its nutrient distribution between water microorganisms that travel through the soil can grow vegetation suitable for animal species development. In order to bring the marshland's benefits to the inside part of the city, we found the proximity relationship between bird location points and green area. With the exploration, we extracted the high-density spots, marking them as the most suitable areas for habitation and reproduction. Due to the slime mold's properties to find the optimal path and its intelligence connecting food sources, the optimal locations were used as an input for experimenting with slime mold as a biocomputer. After testing the locations, a nutrient distribution network from the marshlands to the inner city was created, bringing the marshland properties to the optimal habitat location. The slime mold's new network was then applied into the London terrain, and using a generative adversarial network, we generated an aerobic morphology that takes the marshland properties and redistributes them along London, thus creating the marshland city. A formation that changes with time and height, recreating a marshland typology combined with the human infrastructure. This results in a new type of interaction, whereby providing the non-human the necessary resources to be self-sustained, the bridge to the human infrastructure introduced. This post-natural city shows a 31% growth of green area, but maintains a delimited boundary between entities. The marshland distribution within the city helps to repopulate biodiversity loss by bringing nutrients that microorganisms require in order to grow. After analyzing the marshland city, materials such as algae and bacteria were extracted and combined with cement to form the material that will shape the human and the non-human programs. Understanding the entities' interaction, a fibrous programmatic structure was created with different grids according to its main user. These programs are combined in each extruded volume to create a space for coexistence, allowing each entity to develop its own environment. Each volume becomes a nest that hosts multiple entities with different programmatic activities. With the marshland city and the fibrosynthetic nest, biodiversity can find its properties to grow. At the same time, different entities coexist with a boundary that can reduce human and non-human interaction issues such as COVID-19. The fibrosynthetic nest allows human on the inside and non-human on the outside. The marshland city embraces a flood caused by climate change to establish controlled coexistence in a post-natural scenario, thus creating the biocoenosis nest.
So these basically were, just two visions to try to kind of participate into this conversation of imagining the city of the future. But I think maybe what is important is also to look at the methodology, and somehow I think both in the lab and as in Ecologic Studio, we have always tried to understand the formulation of these visions as the kind of description of a process that often starts from the understanding of the landscape, the reading of the landscape, whether it's natural or urban, or even in this case, sort of post-apocalyptic, if you want, landscape. And then from there, through the use of computer simulation, try to understand how different scenarios may evolve. And of course, again, same as it happens at the scale of the prototype of the small architectures, there is a combination of science and number crunching on one side, and speculative visual element on the other side that kind of drive the project into their scenarios. Claudia, maybe you already mentioned before that there's always been a tight link between the kind of academic teaching, researching aspects of our life and practice such as Ecologic Studio, and of course Claudia, as she mentioned before.
Yeah, 3 years ago I founded the Photosynthetica Consortium that bring together the studio with UCL and Innsbruck, and in particular cyanobacteria and microalgae that, as we discussed in the past, they exist in multiple form, color and shape, and can contribute to reconnect the cycle and restart up the microbiome of the city to eventually create a more complex organism, ecology, and synthetic landscapes as such as the one we've seen before.
Yeah, and some of the recent papers we wrote, which by the way you can find on our website if you're interested. There is a project section and there is the knowledge room section with all the writings you can explore. And they also will come out in the book. We try to talk about this idea of the urban sphere, which kind of connects with some of the concepts we've discussed already in these days, and the idea that of course cities are in a way the centers of maybe urban life, but at the same time they are part of a network that essentially feeds them of the necessary energy, matter, food, information for their survival. And although the world population is growing not as fast as before to a certain extent, the process of urbanization is instead accelerating. And this is again not just people moving to city centers as such, but it's really about people developing a dependency towards this infrastructure that feeds them and supports our life, and kind of losing the ability to sustain themselves essentially. So that kind of leads to obviously the need to find new practices, new methodologies to guarantee this level of self-sufficiency within the urban sphere, but also to effectively reduce the effects, the toxic effects that this type of process, this type of cycle has. And we talk often about remetabolization, like remetabolizing substances, and this again is connected to the idea that cities today have a kind of linear metabolism. We already talked about that. But again, these are I think in the context of new visions, like again we're talking about the line. Maybe the line should be understood as a ring that goes around the whole world and essentially recirculates within itself whatever it consumes or processes. I think we need to kind of push ideas where the whole conception of the city has got at its core, at its heart, this notion of remetabolization, of reprocessing. And that's, again, part of what we have seen in these two initial concepts. The process is really grounded on that idea.
So one of the core elements of Photosynthetica is basically creating an alliance with microorganisms that are able to feed on what is dangerous for us. So suddenly elements like CO2, NOx, PM2.5, PM10 are not pollutants in general, but are elements that feed the building, and in turn you get biomass data, O2, and eventually you feed some of the industry of our city that feed back the human, the city, and interact with the environment, the solar radiation, and the microbiome.
I mean, as designers, we can really actually design every one of these loops and understand what it implies closing them. And I think this for us has been always a kind of opportunity for creativity, and that's, I think, the most important element.
So microalgae are a very old organism. Here, hidden below the S, there is a white dot, and this is a petrol tanker. This is the Baltic Sea. So, and then these are algae blooming. So algae bloom and absorb a huge quantity of CO2 already. But what if, rather than uncultivated forests that bloom on the water bodies of the planet and simply suffocate whatever is below, we can culturalize them, as we like to say, a mix of cultivation and culture, and integrate them in our architecture, making them part of an architecture that becomes alive. Maybe it can grow.
So, I mean, you've seen this process already. Even upstairs, we have this little reactor. The biobomb is a basic unit of functioning. But how that becomes part of a circular economy or ecology within the urban environment. So recently we were able to create this space at the building center in central London. It's near the AA and the Bartlett, and it's a public space you can go and access, and we actually built what is effectively an extension of our studio there, and we regularly spend time there demonstrating how this process can be effectively closed in the urban realm. So we have a set of reactors that in real time are growing algae. So now we also have fresh algae grown in central London. We have 4 species of algae there, Cyanidium, Spirulina, Chlorella, and Porphyridium. And they have different colors, different properties. But essentially what we are able to do is to transform them into biopolymers that can be 3D printed and then eventually become new products. This is a collaboration with Otrivin, which is a brand of GSK, consumer healthcare. So it's about demonstrating how we can turn something that is harmful to our health into biodegradable material and products that can enter the supply chain of a company of that size, eventually eliminating the need for tons of plastics in their supply chain.
This project started 3 years ago, a project that we will see later related to playground and cleaning the air, but then we discussed how this could have other effect in their own organization and in their own way.
We have a little video showing this in motion.
Through the collaboration with Otrivin, we started to imagine a new kind of public realm where the filtration or the purification of air becomes an act of playfulness, an act of interaction with the microclimate that we breathe every day. The Air Lab is really a collection of actions or practices. In the Air Lab, we close the loop and explore what we can do with the biomass that we extract from the purification process. The biomass that we transform into biopolymer first, so a biodegradable form of plastic that is entirely made with renewable and degradable ingredients, and then we transform these into filaments for 3D printing. This process enables us to optimize the position of this material and create products of self-care that are, again, entirely biodegradable, and in this case also carbon neutral. The neti pot is one of these examples that visitors of the Air Lab will also be able to use and practice with. And it materializes this near future dream where people will be able to take care of themselves while taking care of the environment that they are part of.
We are often said that we need to stop the ecological disruption that our planet is going through. But if we think about it, change is an inherent quality of planet Earth. Planet Earth change continuously. We cannot stop change, but we can design architecture, devise architectural instruments to establish a different understanding of change, as well as positive dynamic rather than disrupted one.
So recently we have engaged with another one of our clients, in this case the automotive industry, and in actually scaling up of this process and try to understand if we could make structures of a certain kind and of a certain robustness. And so we developed this project which is called the 3-1, and it's based on a similar material process, but it's engaging with robotic 3D printing and also large-scale 3D printing to take the same material process to a different level of scale. So The tree one is now installed in Busan in South Korea in the Hyundai Motor Studio, and the new year will be in Seoul, and so it will continue its journey. It's a 10-meter tall structure that we understood not as in a sense, a synthetic version of a tree, although that was probably what the client imagined at the beginning, but it's rather for us a demonstration of the opposite process, what we call the naturalization of technology. So the idea of technology fusing back into the biological, into the material world, and somehow all the intelligence that is combined from digital and material processes, biological processes, becomes embodied into the fabric of architecture itself. It's almost like the machines disappear in the background, and the process eventually could become completely biological. We are probably not there yet, but at the same time there is something quite unique about this project. First, that it sustains these living colonies of algae, which themselves become the material to produce another tree, to produce another. So there is a pretty direct closed loop that we can embody, and so that make the system continuously developing. There is we also here have a video that I would like to play to demonstrate a little bit more about this process. So you can see its conception and evolution. The process actually starts from the idea of a column. So although we call it tree one, essentially we think of it as an architectural component, which is again redeveloped and renaturalized. The initial process is digital, as it's often the case in this type of work, and so it kind of treats the typology of the column as a kind of fertile ground to grow these digital fibers, if you want, and that create the structure or the resistant elements. And the process then evolves into a kind of further differentiation of the trunk from the canopy. This was something that became more and more obvious as we were moving on in the fabrication. The trunk being the structural part, and the geometry becomes pleated, as you can see, and so the thickness remains very, very small. Its structure is quite lightweight, but still resistance. In fact, we were able to change nozzle and adapt the 3D printing technology to make the structure more and more thin as we go up. So we start from 1.5 centimeter until 8 millimeter, which is the thinner nozzle we're using on the top. The canopy is an entirely different printing technology because the resolution is much higher, and so we have used 20 large-scale printers, which we have coordinated into generating this type of fibrous structure. Interestingly enough, the whole process has been engineered to create zero waste. So all the 3D print is done in such a way that it doesn't require any support material. Like you see upstairs, you don't have to break off stuff. And that's how the canopy also evolved that shape. As you can imagine, it's printed upside down in the machine, so that it can come up from the ground up and then join into the fibers, then it's turned around in the
original point cloud, as Marco was saying, refers to our Corinthian column that actually is related to one of the headquarters of the client, and then is reorganized in this form of synthetic tree.
So in a way, the process, the project, besides being a kind of sculptural piece mainly, for us is also a demonstration that it is possible to step-by-step scale this type of material technology and effectively generate architectural components and structures that are indeed alive and carbon neutral in their lifetime. So this idea of carbon neutrality, obviously it's the main theme of the architectural section of COP26, and of course this year 27. So with the same client GSK, we developed this project which we brought to Glasgow last year, and it's now traveling to Egypt to become again an element of to kind of instigate, a different type of conversation around the idea of carbon neutrality. And here we have pushed the concept to the extreme, almost saying, okay, if we want to imagine a completely carbon neutral architecture, we need to try to make something that is, in a way, as much as possible made of material that are completely organic and alive. And one option for us was to develop this membrane, which is only 0.2 millimeter thick. So it's extremely thin layer of TPU, which is a thermoplastic rubber, and it's engineered as a three-dimensional matrix. So where the strengths of the architecture comes from here is a combination of the elasticity of the material with the properties of this structure. So the cells are divided into these tubular elements. The air is trapped inside, so it's a kind of seal structure. You can enter as into a tunnel, and within that there is water that acts as a ballast, so it keeps the structure down, because there is about 6,000 liters of water in the base, and also evaporating in the gap creates the diffusion, the diffraction of light that is necessary to guarantee the life of the algae as well as the comfort of the inhabitants.
Interaction and the capability to interact with the environment, and so experience architecture and ecology as a form of pleasure is one of the key aspects of this project or playground. We have designed a series of them. This is a portable one, we have a permanent one in Barso, and so the idea is that by, in this case, bouncing, we activate the recirculation of the air and therefore the extraction of CO2 from the environment.
So again, in this case, the same act of inhabiting the architecture actually contributes to its life cycle and to its performance. Here you see the reactors that in this case are also soft and zipped to the main structure, so everything is also very easily to take apart, and all the material can recycle. Another elements of this project was its augmentation, and we were talking about the other day how to scale up material that cannot be standardized in a way, right? So structures that are by definition changing. So one way is of course to establish a direct feedback, right? So if you can have sensing elements, in this case are electronic sensors, could be also biosensors themselves, right? The biological organisms giving you feedback on their status. Then you can begin to establish protocol where the real-time monitoring becomes the way of guaranteeing the stability or performance of the structure. In this case, it was November, it was Glasgow, we had one day 75 kilometers per hour wind. There was a certain concern that the structure may not hold. So we positioned these 4 sensors each side. They are accelerometer that measure the vibration of the structure and essentially give a feedback, a direct feedback that we actually enabled or amplified with this lighting system, right? So you, on one hand, have an immediate feeling of how much the structure is vibrating, and it's vibrating too much, but at the same time, of course, it just makes it a living system responding to whoever moves inside. So again, the idea of life, it's not confined to the algae themselves, but it's the whole architectural organism composed of its systems that becomes alive. This same technology is a part of a new project that it's still in the kind of a conception phase, but we just wanted to bring into your attention. It's for the ZKM Museum of Media Art in Karlsruhe in Germany, and they asked us to imagine a facade system that would take over the main entrance of the museum and essentially bring the kind of photosynthetic architecture potential to the museum itself. So again, maybe connecting to what Mitch was presenting before, the idea of the facade here that becomes a kind of inhabitable vertical algae garden space and combines elements of delight to the actual performance of a carbon capturing technology.
and allow access to the different level of the museum, while at the same time cultivating multiple species of microalgae that they can then reprocess in the lab that they have in the museum itself in collaboration with the University of Karlsruhe.
So here are some of the drawings we made. Essentially, it's like a very much a kind of system design, and most of the expression is in the kind of membrane that drops in, but also in the, of course, dynamic aspects which we imagine similar to the one of the bubble, but of course in this case at the scale of the entire facade. So this is a kind of a real-time action-reaction that gives us an idea of how much the algae are capturing, what is their health status, and also, you know, the interaction with the public itself that will be moving within the facade of the museum.
This is Ortus XL. It's in itself a one-to-one architectural prototype where algae are inoculated, in this case in biogel. Biogel is a more experimental medium that to contain algae doesn't need liquid water, is lighter and can be itself 3D printed in the matrix in the substratum. But at the same time, for us, this is also a scale model of a larger project that we presented was originally commissioned by the Pompidou, and then when we presented in a later stage, we started to speculate how this would become a scale model of a large project.
Yeah, this was also one of the first main pieces to come out of the Synthetic Landscape Lab in Innsbruck, and really with the collaboration of the team there. I think the idea was really to explore how 3D printing could enable us to create bioreceptive structures, because obviously through 3D printing we can arrange material in layers that are of the same order of magnitude as these microorganisms, and therefore create cells or internal organization where these organisms can grow. And in this case they grow on biogel, but are contained in this matrix that is at the same time open while being receptive.
So in that case, we, for example, similar to the 3.1, we don't have any substratum. Actually, we program the substratum of the Delta printer, and what you see here, this triangulation, is actually the substratum generated to sustain the structure and becoming itself the cell of inoculation of the biogel.
So of course it's a sculptural piece, but it's also understood, as Claudia was mentioning, as a kind of one-to-one chunk of a building, if you want, and potentially a model for
again,
this.
also the hexagonal honeycomb structure that you see there can be conceived as an architectural cell, but at the same time was also, again, the printing plate of the printer that we use at the time.
Yeah, when we imagine this to be scaled up at the scale of architecture, I think there are a couple of things that happen. On one side, at the morphology, this convoluted morphology begin to act as a sponge, let's say, in the urban atmosphere. So the idea is that the building actively becomes a microclimatic agent and essentially mines from the atmosphere the particles of carbon and also pollutants such as nitrogen dioxide and carbon monoxide, etc., that can be transferred into the biomass itself. So essentially remetabolizes them into biomass. The biomass then enters the cycle, the programmatic cycle of the building, feeding its own performances. As you can see, the building, of course, it's open to different forms of life. The idea is, we were mentioning before, to not restrict to only the algae, but also to create possibilities for other species to colonize part of the morphological convolutedness of the skin itself, which again, like in the case of Ortus, becomes open and
it's
part.
argued also in terms of program and contain on the atrium these a tube-like cladding that interface with the labs. And we had the chance to meet a client that allowed us to build a one-to-one version that you had the occasion to visit in Italy of this honeycomb cell, which is the Biotecat that is being built in Astana originally for the Expo, but remained to the city as a museum of biotechnology, so it's a permanent photosynthetic architecture.
Actually, 3 cells. I mean, the example of how 3 of these modules could be essentially operating in real life. And in this project, we try to imagine the 3 different scenarios that were exploring, let's say, the three ways in which we approach nature, essentially. Let's say there was the lab part, which was more concerned with a kind of rational approach, like science-driven, diving into the micro scale of the algae. And then
in that area we produce 1 kilogram of dry algae per day, which is releasing 10 kilowatt of energy. The art space, or the area of interaction, where the user was invited to pump CO2 inside of the structure and interact with the morphology, bubbling, and characteristic of a photobioreactor. Here is a detail. It absorbs 2 kilograms of CO2 per day, which is the equivalent of 32 large trees or a small urban forest. This is the third area, the garden, the production area, where every 2 weeks we have a public harvest and the algae get harvested and transformed. We produce here 600 grams of protein, which is a daily intake of protein for 12 adults, or the equivalent, if it will be meat-based protein, of 8 cow. These numbers are not here to justify the presence of the biotech art, but are there to discuss how number, energy can become material activation, can become architecture itself.
Yeah, again, here the internal organization of the space was very much around these coils of pipes, essentially, which at the same time provided the habitat for the organisms, but also enclosed the space that human visitors were inhabiting during the period of exhibition.
And this brings us to another project that deals with health and interaction, and is the air bubble playground in Warsaw, developed with GSK. Warsaw is one of the most polluted cities in Europe. The client wanted to develop a set of urban activators that will deal with the respiratory system, especially of young adults. To start, we can see a video from such and such in London of sensibilization on the topic.
Yeah, it's part of the campaign run by such and such.
Stacked in the middle. Do I send them out to play collision or keep them locked up inside?
Is a major problem. Pollution levels are far worse at the child's height. They are more sensitive to the detrimental effects of air pollution.
We all grew up going outside and not having to worry about things.
I never thought I'd have to protect my kids from the one thing that keeps them alive.
As a mom, it breaks my heart.
I just want to play.
Microalgae are incredible organisms. And with our technology, we can use them to clean our city's air. We have gone to one of Europe's most polluted areas and built the world's first air-purifying biotechnological playground.
The uniqueness of this concept relying on the relationship between the architecture and the kids.
The more children play, the more the algae absorb pollutants and clean oxygen is released.
This algae is what's taking the bad stuff out of the air.
The air feels amazing.
The key aspects of this project is to create awareness that it is indeed possible to clean the air we breathe.
This can't be the only way to help them breathe better. We need to do more.
Well, one of the main challenges with the project really was when we had meeting, and we had quite a long time of discussion on how we actualize the project, was mainly to explain how it was the architecture that was interacting with the environment through the ETFE membrane and its morphology, and the bouncing of the kids that would allow the remetabolization of the air. Because most of the questions were going towards the technology. Is the technology tested? Does it work? I don't know. Did you test your grandma house before you built it? The question is that it was really a challenge for us to discuss how in architecture we run simulation, we run tests at different scale, but we don't build an architecture before building it and testing it. We work through simulation and drawing, and it's the architecture itself that contributes to the urban environment. It's not an architecture with attached technology that will save the planet. Yeah.
Yeah, I mean, I think in this project, maybe I go, yeah, exactly. I think in this project is really the way we try to arrange the different layers of the system that makes it, in a sense, performative. So, of course, there is this ring of reactors, which is the active principle, if you want, where the cultures are located and where the air is purified. But at the same time, what was important for us was to be able to control the microclimate to the extent in which it still felt like being outdoor, but at the same time the pollutants could not enter and basically, let's say, annihilate the effect of the algae. So this was the role of the membrane, the ETFE membrane, and the roof cone itself. One other element, of course, is the structure. This is CLT cross-laminated timber structure. And the interesting thing is that not only hold the piece together, but also acted as a shading elements for the interior and for the algae culture itself, protecting the reactors and guaranteeing the robustness of the system. So it's a whole combination of these elements that effectively make it capable of surviving in the environment. We had
a set of sensors in this case integrated in the playground, outside the playground, and across the city of Warsaw to measure the effect on their metabolization, and we had a reduction of PM2.5 between 75 and 95% every week in the area immediately surrounding the playground. And also the aim was to reduce the AQI, so the air quality index from WU, inside of the green area in the area immediately surrounding the playground in comparison to the rest of Warsaw.
In this case, the target was towards what the World Health Organization recognizes as levels of concentration of pollutants that are suitable for health, and our objective was to make sure that within the bubble we could achieve that concentration, or reduce the external concentration to that level. As we were mentioning, microalgae also have a nutritional value, especially algae like spirulina or chlorella. They are already part of the food system, and when consumed fresh, they actually have an even more powerful action. So one of the ideas that we have explored since the beginning was how to bring fresh cultivation of these organisms within the built environment. And then, of course, in our attempt to push this to the next level, we accepted to engage with Nestlé in this collaboration. They came to us asking if it was possible to test this system within their supply chain. They already have algae-based products, although they are very much marginal to their whole market, but of course there was the intention to do more. So as a first test bed, we installed this array of reactors in their headquarters in Lisbon, and again we tried to make it as much as possible part of the facade of the building, and therefore to the architectural system. And again, this is something that we try to insist every time to make sure that it is understood that the idea is not simply to just bring another technology to the table, but rather to really induce new behaviors within the architectural experience of the space. And then, of course, these are some of the concepts we are developing with them about the idea of a biofactory. So where essentially factories that in this case produce food grow as much as possible of the raw materials of the aliments on the building facade itself, on the envelope of the factory. So let's say the idea of the big shed becoming a big garden, if you want, and then all of a sudden through the help of digital technologies, in this case also augmenting the possibilities of the production line towards essentially as short as possible supply chain, ideally a completely closed one. I think we are still a little far from that, but it's a first step. And it's very interesting that these type of companies are engaging with these processes and are testing these solutions.
This is a concept that we engage with also in the latest Venice Biennale. The question posed by Asim Sarkis was how will we live together, and we start to envision how do we live together with microorganisms, and we devise a prototypical home unit, whereas composed of 3 elements, the filtering system that are the curtain, the growing system that it's the vertical garden, and the convivium, which is the area of consumption, where we consume the microalgae itself. Here you see a vision of the garden and cultivation as we imagine it, and here there is a vision of the convivium, the area of experience. We develop a set of glassware, 36 3D printed glassware with Swarovski, each of them containing a medium of microalgae, potentially having the same amount of protein of a large sack. We couldn't consume the algae in that case because unfortunately there was a pandemic, so we couldn't serve the food. That was not possible, and that's a type of prototype that we have also extended to the home use. We have one
unit upstairs, so you know.
We have a unit upstairs,
how
not
this
containing spirulina in that case. I give you, so I will not advise you.
Yeah, don't bring.
Don't bring the one upstairs because the same medium is not for food.
Now this is a simple piece, but I think what we tried to do with this was to actually develop the sort of simplest and most, I would say, elegant possible unit in a way to try to propose an idea of a product or interior design product that can realistically sit in people's houses and at the same time be an effective way of growing this organism, and especially for daily food consumption. I mean, we have been testing it in several locations. We have it at home and in the studio now since a couple of years, and it's working. It's a system that we keep tweaking, but it's essentially working quite well. And in the end, the idea is that it's made essentially from a glass vessel, which is the chamber for the algae to grow, and the 3D printed components and bioplastic are all dismantleable, and it's very easy to separate and recycle every single piece. So again, it's also about thinking of a design that can be either composted or recycled without problem from anyone, like even at home. So maybe we should conclude. I think it's 6:30 now.
Yeah.
Right? But yeah, I mean, essentially, I think just to conclude it, we wanted to kind of resume out at a sort of urban scale, just to kind of touch the question of what happens when these type of visions are implemented and buildings become essentially able to grow their own resources and able to synthesize material and energy. I think we can begin to imagine the city not so much as a kind of centralized set of infrastructures like it is often the case today, where essentially the source of energy or the processing of waste happen in one place, but it becomes a rather more distributed notion. And I think again, when we are normally exploring visions for future cities, this idea of distributedness and distributed intelligence, it's something that is very present in the back of our head. And so we try to always map how that would look like in the real scenarios. This is a project we did for the city of Tallinn in 2017, when Claudia was a curator of the Biennale there, and then we continued the process. It was as a sort of blue-green plan for the city that would look at what happened in 30 years. The city is as you know, in the Baltic Sea, and it's surrounded by water, and actually has there is a serious prospect for it to be flooded as the level of the sea rises. So that's a very clear reality for them. The project started from a very technical analysis of the landscape, the urban landscape system infrastructure, and then in a particular area, which is the Palasar Peninsula, we extrapolated a more visionary element of a kind of new extension of the city that becomes the kind of metabolic alter ego of the existing one. So in a sense that takes the wastewater from the main city and remetabolize a new landscape, producing resources for the existing one. Same approach into this collaboration with the United Nations Development Program. Again, here it was more to develop a toolbox, in a sense, for architects, for other architects, and for public administrators to deal with cities like Guatemala that have a very high percentage of unplanned development. So you know, like more than half of the city is developed informally, if you want, or let's say outside of the planning code. And essentially that offers the necessity for imagining infrastructures that can deal with the excess waste, that can deal with the lack of water, that can deal with all this instability of the soil, and within that logic. And that's what the tool set does. Claudia, you're going so fast that I can't even breathe. But no, I mean, it makes sense because I guess we are all exhausted of hearing people talking. So, well, unfortunately, we were the last one, so you're gonna have to bear with us. But no, I mean, we are at the end, and again, this is the creature that you've seen upstairs. And actually, we're trying to revive it, but it's struggling a little bit. When it's alive and kicking, it creates these amazing networks. We've always been fascinated by this. We're not the only one. It's such a weird organism, the slime mold, that even scientists are not sure where to place it in the big charts of organism, because it's a single cell organism, a protist, but it's made of many nuclei that flow. Effectively, you can understand it almost as a sort of water infrastructure, or that is able to modify its geometry in its body and kind of reach out in many different conditions.
From the last exhibition, Mutation Creation of the Center Pompidou, that was titled The World of Network. Actually, the slime mold even got the main poster in this case. We were asked by the curators to develop a project about Paris by interfacing slime mold and artificial intelligence.
So we also made a biopainting which is now in the permanent collection of Pompidou. So I'm very proud of that because slime mold is in the permanent collection of Pompidou.
Don't tell them that it can be revived. So the biopainting and the computational videos we acquired by the Pompidou thanks to one of their donors, Chanel, who was also very happy to hear there were women in the team despite the subject being computation. And so these are some slides from the computational video. So we train an artificial intelligence algorithm to rethink Paris from a scale of 10 kilometers to the scale of the Pompidou, and transform the Haussmannian Paris and their boulevard in a more deconstructed blue-green city. And here we are zooming in scale. And the next is my favorite scale, is the scale of the Pompidou, where the algorithm is basically reprocessing the infrastructural system of the Pompidou itself in a sort of wet one.
We just wrote an article about this project that is going to come out very soon in the next number of AD, and it kind of articulates the project and the sort of philosophy behind it, also in relationship to another kind of utopic vision called New Babylon, which I don't know if some of you know already. It's a story of a city where basically systems and infrastructures have taken over the hard job and allow people to essentially play and engage in fun activities and essentially go back to a form of nomadic existence within the infrastructure of the city itself. And it was very much inspired by some of the situationist approach to the city, the idea of psychogeography of the city. And it's a very interesting, I don't want to bore you too much because it's quite late, but we have been writing about that, and I think there's a kind of connection could perhaps also inspire you in some of the work that we will do in the future, so you can go and check that. Mark Ridley, I think he wrote a lot about New Babylon, so there is a whole history there about this kind of idea of a network city that is somehow infinitely extended and adjustable. But the video probably you already seen it to the first day, but maybe it's kind of cool to close the loop and see again the end. And it zooms through the scales, so it kind of engages with essentially with the satellite image of Paris and reorganizes it through the, let's say, behavior of the slime mold. And this happens at different scales. And I think what is interesting is that when you zoom in, you know the algorithm reads different patterns, reads different kind of infrastructural element. The first is 10 kilometer. It's really to do with infrastructures, because that's what you read at that scale. That's what the satellite picks up at that scale. And then zooming in, you begin to deal with neighborhoods and blocks. And then obviously the great frame when you reach the scale of the Pompidou itself, which is a big machine, it's a big architectural machine. Kind of reads the pipes and kind of rearranges them into a kind of bioorganic form. And I mean, if you think about the Saint Pompidou within the center of Paris, the shock that created when it landed there, and the fact that it was made possible, I think that the jump of imagining an entirely bio-digital wet architecture, it's not that far. So the problem is that to find brave enough missions these days. I think the Pompidou itself was a very unique process. But you know, these things happen. So anyway, that's it for today. And thank you for your patience.
Thank you.
Long day again, but hopefully, I mean, surely.